Welcome students, I am Atin Debinsha, lecturer of Human Health College of Pharmacy. Today topic gel electrophoresis. It is one of the type of electrophoresis. First we see the introductions about electrophoresis. We already see in paper electrophoresis. Once again we see the electrophoresis definitions. Electrophoresis is the separation of charged molecules in an applied electric field. It helps to separate the charged molecules by using the electric field, that is called the electrophoresis. The relative mobility of individual molecule depends on several factors, the most important which are net charge, molecular shape, the temperature, porosity, viscosity of matrix through which the molecule migrates. Next we see the types of electrophoresis. The electrophoresis classified into two types. One is zone electrophoresis, second one is moving boundary electrophoresis. The zone electrophoresis divided into four, that are gel electrophoresis, paper electrophoresis, thin layer electrophoresis, cellular acetate electrophoresis. Again, the moving boundary electrophoresis divided into four, that are isophoresis, capillary electrophoresis, isoelectric focusing, immunoelectrophoresis. These are the types of electrophoresis. In this term, we see the zone electrophoresis type, that is gel electrophoresis. Next, we see the introduction definitions about gel electrophoresis. Gel electrophoresis means the size exclusion gel electrophoresis. The gel electrophoresis is a separation technique in which separations of compound based on the molecular size by using the supporting medium of gel, that is called gel electrophoresis. The separations of compounds based on the molecular size by using the supporting medium of gel, that is gel electrophoresis. This technique is used for the separations of DNA, RNA or protein molecules according to the molecular size and the electric charge using an electric current applied to the gel matrix, that is the gel electrophoresis. Gel means what? Gel is a cross-linked polymer whose compositions and porosity is chosen based on the specific weight and porosity of the target molecules, that is gel. The different types of gel we use in gel electrophoresis, that are agarose gel, polyacrylamide gel, and aluminum and peptide gel, Sephardex gel. These are the gels we use in the gel electrophoresis techniques. This is the structure of the gel electrophoresis. Next, we see the principles of the gel electrophoresis separations. Their principle is based on the charge and size. First one, according to charge, when charged molecules are placed in an electric field, they migrate towards either the positive or negative, anode or cathode pole according to their charge. That is charge based separations. Second one, according to size, the smaller molecules move more swiftly than the large size ones, as they can travel through the pores more easily than the latter. This is the principles of gel electrophoresis. Next, we see the gel types of gel about agarose and polyacrylamide gel. First, we see the agarose. Agarose is a polysaccharide extracted from seaweeds. Gel cast horizontally. This agarose gel is non-toxic and it separates large molecules commonly used for DNA separation. Agarose staining can be done before or pouring the gel. Second type, polyacrylamide gel. It is a cross-linked polymer of acrylamide. Gel cast vertically, that is horizontally, this is vertically. It is a toxic, separate small molecule. That agarose gel is separate large molecule, but polyacrylamide gel is separate small molecules. used for DNA and protein separation. The staining can be done after the pouring the gel. This is the difference between agarose gel and polyacrylamide gel. Next, how do prepare the agarose gel or preparation of agarose gel? Take agarose powder and mix with distilled water, and next add buffer solution for maintaining pH, and add small amount of agar and place it at room temperature for solidification. Then uh, we produce the agarose gel. This is the preparations of agarose gel. Next, instruments of gel electrophoresis. First one, electrophoresis apparatus. Second one, buffer. Third one, power supply. Fourth one, supporting media. Fifth one, detection and quantification. These are the instruments we used in the gel electrophoresis. First, electrophoresis apparatus. The casting tray is made up of glass or plastic. The electrophoresis apparatus is made up of glass or plastic. The comb contains varying number of teeth in order to help in formation of well. That is electrophoresis apparatus. Second, apparatus. The electrophoresis apparatus setup. This is the electrophoresis apparatus setup. It contains casting tray, electrodes, electrophoresis chamber with buffer solution. Second instrument, buffer. Buffer in gel electrophoresis are used to provide ions that carry a current and to maintain the pH at relativity constant value. Why the buffer is used in gel electrophoresis means to provide ions that carry the current carry and maintain the pH. The most common being for the nucleic acid. What are the buffers we use means TAE and TBE. TAB means tris acetate EDTA and trisborate EDTA. This is the buffer. Third instrument, power supply. What is mean by power supply means the electrodes are connected to their respective terminals of the electrophoresis chamber and to the power supplier with the controls for rate of current flow. The best resolutions of fragments larger than about 2 kb is attained by applying no more than 5 watts per centimeter to the cell. This is the power cell. The main parts of the gel electrophoresis power supply. When electric current only flow means the charger particles are moved, migrates, that cause separations. It is power supply. Fourth instrument, supporting media. In the gel electrophoresis, we use the supporting media as a gel. And many gels we use. First one, starch, agarose, cellulose acetate, polyacrylamide gels. These are the gels we used in the gel electrophoresis. The kind of supporting matrix used depend on type of molecule to be separated and the desired basis for separation, charge, molecular weight, molecular size, etc. We separate the particle we use in the supporting media of as a gel. Last one, detection and quantification. The stains and blotting we detected by using the gel electrophoresis. Stain, protein staining, ethidium bromide staining, blotting, southern blotting, northern blotting, western blotting. Southern blotting for DNA determinations, northern blotting for DNA RNA determinations, western blotting it is used to treat determine the protein. These are the detection techniques and quantifications. This is the instrumentation of gel electrophoresis. This is a ethidium bromide stain and the protein stain. This is the structures of the detection and identifications. Next, this one is the process of gel electrophoresis. How the gel electrophoresis done. First we play gel is prepared and the buffer will added and the sample is dotted and the current will apply the based on this charge particle based on the molecule size weight we separate the particles. This is the process of gel electrophoresis. Next one, factors affecting separation in gel electrophoresis. What are the factors affected gel electrophoresis mean? First one, sample. Sample is affected by gel electrophoresis. The charge ratio of sample dictates its electrophoretic mobility. This the charge size shape. The higher the charge, greater electrophoretic mobility. But lower the charge, less electrophoretic mobility. So the sample affect the gel electrophoresis. Next, the size, sample size. The size is inversely proportional to electrophoretic mobility. So sample size is affected by gel electrophoresis. Next, sample shape. The globular substance move faster than the fibrous one. So the sample affect the gel electrophoresis. Second factors, gel. Agarose and polyagarose gel are affected the gel electrophoresis. And the agarose gel have very large spore size and are used for separating larger DNA molecules. So agarose gels affect the gel electrophoresis. The second gel, polyacrylamide gels are used to obtain high resolution separations for smaller DNA molecules. So these polyacrylamide gels is affect the gel electrophoresis. Agarose and polyacrylamide gels are cross-linked sponge-like structure. It is important that the support media is electrically neutral and the presence of charged group may cause migration retardations, the flow of water towards one or the other electron, so called electroendoosmosis EEO, which decrease resolution of the separations. In this paragraph we define the agarose gel and polyacrylamide gels affect the gel electrophoresis. Next, the buffer. These buffer components also affect the gel electrophoresis. The buffer can affect the electrophoretic mobility by ionic strength and pH. First one, ionic strength. Increase in ionic strength of buffer means the larger share of current is carried by buffer and the smaller portion by sample, while decrease in ionic strength is vice versa. So ionic strength of buffer affect the gel electrophoresis. Second one, pH. pH determines the degree of ionization of organic compound, where ionization is inversely proportional to pH. So pH buffer pH also affect the gel electrophoresis. These are the factors affecting separations in gel electrophoresis. First one, sample, next gels, next buffer. Buffer classified to ionic strength based and pH. These are the factors affecting the gel electrophoresis. Next one, advantages and disadvantages of gel electrophoresis. First one, advantages. First point, stable chemically cross-linked gels, and second point, greater resolving power and good for separations of low molecular weight fragments. Pore size of acrylamide gels can be altered in an easy and controllable fashion by changing the concentration of two monomers. And next one, the gel is low cost and easy to operations. This is the one of the advantages. It is a time consuming technique. And second, disadvantages of gel electrophoresis. Generally, first point, generally more difficult to prepare and handle, involving longer time for preparation than agarose. So this one is prepare more difficult. Second point, need new gel for each experiment. Each experiment we need new gels. So disadvantages in gel electrophoresis. Next we see the applications involving gel electrophoresis. So, gel electrophoresis is used to separate the deoxyribonucleic acid. Second one, separation of ribonucleic acid. Third, separations of protein molecules. Fifth, it may be used as preparative techniques prior to use of other methods such as mass spectroscopy, cloning, DNA sequence, southern blotting for further characterization. And next, the separations of amino acids. It is used to separate the lipoprotein. It is used to separate of enzyme in blood. It is used to separations of antibiotic drug. Next, to analyze results of polymerase chain reaction. The polymerase chain reactions is analyzed, and particular gene associated with a particular illness to be analyzed in DNA profiling for taxonomy studies to distinguish different species. In paternity testing using the DNA fingerprinting. Electrophoresis is used for DNA testing, fingerprinting testing. In the study of structure and function of protein, in the analysis of antibiotic resistance, in blotting techniques for analysis of microorganisms. It is used to determine the larger molecules and small molecules of DNA RNA substance. These and other advantages applications are is very useful technique in various fields for genetics we use, biochemistry to molecular biology, and other research field we use the gel electrophoresis. Next, it is also useful for the detection of protein expression, purification, protein analysis means cell proteins expression. Other technique we use such as Western blotting, 2D gel electrophoresis, etc. These are the application involving gel electrophoresis. Today we discuss gel electrophoresis. Gel electrophoresis means separations of components by using current. Separations of charged molecules in applied electric current with the supportive medium of gel. That is gel electrophoresis. Next we see the introductions about electrophoresis and types. Next we see the principles of gel electrophoresis, differentiate between the agarose and polyacrylamide gel. Next we see preparation of agarose gel. Next we see the instrument and reagents. What are the instruments we use in the gel electrophoresis that also see today. Electrophoresis apparatus, buffer, power supply, supporting media, detection and quantification, that's also see. Next we see the process of gel electrophoresis. Next we see factors affecting separation in gel electrophoresis. What are the factors affect the gel electrophoresis? That are sample, gel, next the buffer. These are the factors affecting separation in gel electrophoresis. That's also see today. Next we see the advantages and disadvantages of gel electrophoresis. The main advantages, it is low cost and easy to operate, good for separations, great resolving power, stable. This next disadvantages means need new gel for each experiment, and generally more difficult to prepare and handle. This is the disadvantages. And next we see the applications of gel electrophoresis. Many application in the pharmaceutical, genetics, proteomics, and research. These are the applications. we see in this day. Thanks. Thank you.